Good morning, everybody. Uh, we're going to continue to discuss stem cell biology. And today, the discussion is going to get a little bit more technical. And we're going to discuss how to detect, identify, and isolate stem cells. And in the previous lecture, we learned that based on the location of the isolation of stem cells, we can categorize the cells to stem cells in the embryo and stem cells in adult. We can say adult stem cells, but I would rather not say embryonic stem cells to divide these two categories because embryonic stem cell, as we know, is a special term that's reserved for a particular cell type. So next week, we're going to be dealing with entirely embryonic stem cell. So, embryonic stem cell, as I said, it was from the inner cell mass. At the blastocyst stage, embryo, you take the cell out. So you don't have much trouble. You won't have much trouble identifying those cells and get them isolated. But adult stem cells is a different story. They are often located somewhere in adult tissue, and you do have the challenge to identify the cells, and if first you will have to identify them before you can get them isolated. Sometimes you don't even need to get them isolated if you're only interested in how the cells behave in vivo, that means in animal. So on the first slide of today's lecture, I give you an example of adult stem cell. So on this slide you see a section of the brain. So I'm trying to tell you the location of neuron stem cell. Neuron stem cell is a kind of adult stem cell. They are multipotent and not pluripotent. They can give rise to all the cell types in our body. They can only give rise to some cells that's related to the neuron system. So usually we know that neuron stem cells can give rise to neurons, oligodendrocyte, and astrocyte, 3 cell types. So this is by experience. The people have already defined that the neuron stem cells can at least differentiate toward these 3 cell types. So in the future, if you want to tell somebody that you have a neuron stem cell identified, you better show them these cells can become the 3 cell types shown on this slide. But for the purpose of today's lecture, I want you to pay attention to where you find the neuron stem cells. They are located essentially in 2 regions of the brain. One is called the subventricular zone. It's over there, subventricular zone, and the other is in the hippocampus, like over there. So there are 2 regions that you may be able to detect these stem cells. And on the bottom picture here, the green labeled region is the that's the part of the dentate gyrus. So this is the granular zone. It's called the granular zone here. It's the green part, and then underneath that green layer, you can see some blue cells, very small amount, some blue cells in that place. That is the stem cells, neuron stem cells that you will find in the hippocampus. So sometimes we say the stem cells live in a niche. That's a very special environment that will allow the cells to keep their identity. So there will be a lot of times in quiescence. That means they are not dividing. They just live there. Their environment is protected, so they're not dividing, but they are a stem cell nonetheless. They're a stem cell. When the condition will allow, they will start to proliferate. They will replicate themselves, and they will start to differentiate. But according to what we have learned in the previous lecture, the 2 criteria is fulfilled. So they can self-renew and they will differentiate. So that's a stem cell there. So they live in a special environment in most times in adult tissues. So that's one example, the neuron stem cell. And then on this slide, I'm going to introduce to you another adult stem cell type, that's cardiac stem cell.
Professor,
yes, you can ask.
Can you please go back a little bit?
Sure. Yeah, that's. You have a question here?
No, I just want to write something, but thank you.
Yeah. You should be able to download the PPT. It's in class materials in Teams as well as in Blackboard. Upload. Okay. So on this slide, I would like to tell you another stem cell, adult stem cell type, cardiac stem cell. But here, my focus will be, using this as an example to show you how to identify the stem cell. But first of all, let me give you a brief overview of the cardiac myocyte. Cardiac myocyte is the muscle cells in heart. Okay, our heart is just a muscle. I think maybe some of us have the opportunity to consume animal heart, right, as a food source. And it's muscle. It's just like the skeletal muscle. It's just mainly muscles. You will have smooth muscle cells there as well as endothelial cell. Those are major components of the vessels, right? Smooth muscle cell and endothelial cell are the major cell type of vessels. But other than that, most cell will be cardiac muscle cells in the heart. But one thing that's unique to heart cell is, like in humans, there are 2N. At the born, it's 2N, but once they reach adulthood, the cells will be in 4N. Most of our somatic cell is 2N. That means 46 chromosomes, right? 23 from dad and 23 from mom. But the adult heart has 46 times 2. That's 92 chromosomes there. The reason is because they get a rest during cell division. So when at birth, there are 2N, but then they try to divide again. But that cell division will not finish. And then they get arrested at some point, they become 4N. That's very unique for cardiac myocyte. There are 4N, two copies of the genetic material than other cell types. But in mouse, it's kind of similar. In mouse, they also get 2N plus 2N, but they get arrested at a different stage. That's why on this slide you see in humans labeled as 4N, but in mouse it's 2N plus 2N. Basically, they are arrested at different stage of the cell cycle. But anyways, for both human and mouse, you have 2 copies of the genetic material. But in some lower species, like zebrafish, the cardiac myocyte is very similar to other cell types. There are 2N. So that's just one information about cardiac muscle cells. And because of that genetic material difference, or shall I say related to that, the zebrafish heart cell actually can proliferate. There are 2N, they can, replicate to become 4N and divide it to 2N again. But unfortunately, for both human and mouse, they get permanently arrested somewhere in the cell cycle, and they won't be able to divide again. So, yes.
But wouldn't, like, for instance, like having twice the genomic material be like a big issue in the cardiac side?
I think maybe that's, in our heart, the heart cell is so-called the terminally differentiated cell.
Yeah.
And in a previous lecture, you learned that there is not much cell replacement, right?
Yeah.
So you're born with certain number of cells, and that's all you have.
Yeah.
And then they are going to perform the function for the lifetime. So they need a good protein manufacturing ability in there. So you have twice as much genetic material, and I bet you're going to have twice as much genes transcribed, right?
Yeah.
And then you can manufacture a lot of proteins. Maybe that's one of the purpose.
Okay, that's
Yeah, to have a lot of opportunities to manufacture the needed proteins to support the function.
Yeah. Wow, that's pretty cool. Thank you.
I guess maybe that's one of the reasons. Okay, but the cost is you don't have the opportunity to replace the damaged cells any longer, because every cell is now in arrested status. They cannot divide further. Therefore, a major interest of research is try to find stem cells in the heart, because naturally maybe the function of the stem cells are not sufficient to provide the capacity to replace cells, right? But if we can find some adult stem cells in the heart, maybe we can take the cells out and somehow expand them in vitro, so we can have a bigger number. And in case of somebody have a heart injury, maybe we'll be able to inject those cells, and then they differentiate toward cardiac myocyte, and then help the patient to recover, the damage to get repaired. So that's the promise there. So it is a very hot research field. But like I said, today's focus, the theme is how to identify and isolate the stem cells. So you can view this slide as a good summary of the many methods we will discuss in today's lecture of identifying and isolating stem cells. Starting from the bottom left, you see something called cardiospheres. So basically, in this technique, what we're trying to do is get a heart minced, get them cut into pieces, and then you can make a single cell suspension from there. You can incubate the tiny pieces with certain enzymes, such as trypsin or collagenase. Those enzymes can dissociate tissue. And you can make a single cell suspension, and then you culture the cell in certain condition. And sometimes you can find spheres. So some of the cells will automatically generate spheres in the culture. And if you isolate those spheres, and you have a higher chance of getting stem cells. So sphere formation is one character of most adult stem cells. So if you have the right condition, you put them into culture, they turn to form a sphere. Adhere to each other, proliferate, and then form a sphere. So if you get the sphere formation, you have a chance of enrichment of stem cells. So I'm not saying that all the cells in that sphere is adult stem cell, but you just enriched them. The concentration will be higher than just the heart itself. So that's one way to enrich stem cells, is making them to form a sphere. And then on top of this slide, top right, I have something called site population cells. So you basically, the stem cell will have some unique function, like they will have some unique channel on their cell membrane, and you can use that particular unique feature of stem cells and then use some technique, to isolate them. So I will have a detailed discussion about the site population later, but for the time being, site population is a type of stem cell capturing, or the site population is the stem cells. They are called site population because the circular region there, the circular region is on the side of the main population. That's why it's called the site population. Later on, we're going to have a good discussion about that. And then we can rely on the markers. On the cell surface, there will be some markers, some proteins on the cell surface. By detecting those markers, you would be able to detect where the stem cells will be. Okay, basically we have 3 categories of stem cell identification techniques. One is based on their unique culturing feature. And the second is about function. They have specific function. You can utilize that function to identify them. And third will be markers on the cell surface. Okay. And first, I will discuss the use of combination of cell surface antigens. So let me jump to the next slide to first introduce to you some nomenclature system, which is called the cluster of differentiation, or CD. CD basically is a designation of the designation system for cell surface proteins, and those proteins must be able to get detected by established antibodies. So if you have a protein, but there's never any antibody designed against that protein, and then they don't belong to the CD system. If we say this cell surface marker is CD42, that means, okay, this is one of the protein that can be a differentiation marker of the cells on the cell surface, but also that marker can be detected by some commercially available antibody. So basically, there is an international organization. They name the cell surface proteins by give them a number, like CD1, CD2, CD3, CD4. So the cell surface protein will have their original name, like c-Kit. c-Kit is a name, a cell surface protein, and this organization will rename c-Kit to be CD117, for example, right? So, it's just a nomenclature system. So back to the previous slide, I want to tell you what's the principle of identification of a certain cell type by using the CD system. So basically, on this slide, you see 2 lineages of cells. So the AGD cell shown on the top will eventually give rise to 2 cell types, and then that 2 cell types will further differentiate to give their lineages, right? So we have 2 major lineage, and each of those one will give rise to 2 sublineage, and they can further differentiate. So sometimes we call differentiation to be a tree. So you start from the same ancestor and then branch out, and each branch will further branch out, right? So that's how differentiation works. And now you have 2 major lineages. They express different cell surface markers. Like the first group will express AGD, while the bottom will have HGD. So apparently, the surface marker A will be able to help you to distinguish these 2 types, right? The same for H. So if you can detect A antigen on the cell surface, basically you can say, okay, you have the top cell lineage instead of the bottom cell lineage, right? Because A is not expressed at all at the bottom cell lineage. So the A will serve as markers to distinguish the 2 cell types. Sometimes it is not, you don't have the opportunity to detect a cell lineage by just using one marker. It's not possible because they don't have any one thing that's unique for them. Then you will have to use combinations. Like, I don't know if we have an example here. Yes, like now let's focus on, oh, okay, I should point here. So now let's see this 4 cells. You can see you can use A and B combination to distinguish the top lineage from the lineage here, right? Because AB is unique for these 2 cells only. not for anything else. So you can use combinations of cell surface markers as well. If you don't find one protein that's unique, and then you can use a combination that's unique for cell type. And sometimes you can even use a negative selection. That means you define a cell lineage by the presence of the marker and at the same time, the absence of some other markers, right? That is very straightforward. So that's the principles of cell identification by using combinations of the CD markers. And on this slide, I give you, another example. And this is from Libome company webpage. They have summarized the usefulness of CD molecules, CD proteins, in defining cancer stem cells. So this is something new. The cancer, you can consider cancer as a kind of adult tissue. Basically, there are some bad adult tissue, but they're adult tissue. In cancer mass, you still will have some cells that are considered a stem cell. They're not the same as the rest of that cancer mass. They're unique cells. They are the extremely bad guys there, because if you use chemotherapy to kill the rest of the cell, they are going to proliferate. They will self-renew, and then they will differentiate and give you a new cancer. Okay, so that's the definition of cancer stem cell. Still fulfill the criteria of adult stem cell, self-renewal and differentiation. Okay, so those are the target for the clinicians. They try to capture the cancer stem cells. They try to target those cells. They want to kill those cells, because killing other cells are not going to be sufficient to completely remove the cancer, the problem of cancer. So here on this slide, we have quite a few organs here. These organs all could develop cancers, and then those CD markers, the combination of CD markers will tell you how to, what combinations should you use to identify the cancer stem cells. So this is just one example of how we are going to use the CD markers to define a stem cell type. And here, I give you an example. On the left is called tumoral spheres. Sphere formation, one way to capture stem cells, as I explained for cardiac stem cells. You can now, in this example, to identify cancer stem cell, you just make a single cell suspension from a tumor, and then you allow the cells to be cultured in suspension, and some of the cells will form a sphere, like shown here, the sphere. And now in those spheres, you have a higher likelihood to have stem cells in there. And then on the right, it is another example, but in this example of identification, we use surface markers. As you can see, we use a combination of positive and negative selection. So these stem cells are defined as CD44 positive, but CD24 negative. Okay, or low, CD24 negative or low, and they should be lean negative as well. So what is lean? Lean is a lineage marker. Lineage means, when the stem cells get differentiated, those differentiated cells also express some cell surface marker. The combination of those markers oftentimes are summarized as lineage markers. It's not the stem cell marker, but the differentiated cell marker called lineage marker. We say lean negative. So the stem cells will have a stem cell marker, CD44 positivity, and then there should be negative for a bunch of other markers. So we use a combination strategy. But here I want to show you this flow cytometry result. So on one dimension, the horizontal dimension, you have a PE staining, which is CD24 PE, and on the vertical dimension, you have a CD44 APC staining. So as you can see, this group, the red box, the cells in the red box, they're high in CD44. You divide all the cells to four quadrant, and then you see CD44 is high. The cells are high in CD44 but low in CD24. On the horizontal dimension, you move to the right, you have increased the intensity of the fluorescence, and you from bottom to top, you also have increased fluorescence intensity. So the cells are higher in intensity of CD44 but low in the intensity of CD24. So that's that fulfill your definition of the stem cells. You get them isolated.
Dr. Liu, could you explain then what is the quadrant next to the orange box? That's big. Yeah.
Next to the orange box, to the right.
Yeah. Right,
so those cells, as you can see, they are high in CD24 and also high in CD44.
Mm-hmm.
So although they fulfill the criterion of high in CD44, but they're also high in CD24. That means maybe those cells, let's say maybe CD24 is a differentiated cell marker, right? So you want them to be low. And now you see those cells, they're high in CD44, maybe they're a stem cell to begin with, but they have already differentiated somewhat.
Okay.
You don't want them any longer, right?
Yeah.
Okay, so that's, I'm glad you asked the question. Please, just interrupt me. We have a small class. Just
why CD44 considered stem cell, not CD24?
That's because it's sometimes there's nothing to explain, so that they just have markers on their surface that give them different identity. Of course, those markers also have some functions, but for the purpose of identification, we don't really care about their actual function. I hope that makes sense, because there's no reason why one is a stem cell marker while the other is not. It's just a fact. Okay. So,
could you explain the Lin negative concept again?
The Lin negative concept.
Yeah.
So the Lin, the name Lin, oftentimes is used for the hematopoietic cell lineage. We say something as a lineage marker. That means, for example, in hematopoietic cell system, you have a lymphoid lineage, you have a myeloid lineage. So they are the two branch from the original lymphocyte. You have two branches. We call this is lymphoid lineage, while the other is myeloid lineage. That's just one example. And then later on, you can say let me come here, I can explain to you. Okay, so if these two cell types are from the same origin or ancestor, and you say this is a blue lineage, for example, and this is a brown lineage, for example, and then you have an AB lineage that's here, you have an AC lineage that's here. So the lineage is, based on the meaning of lineage in the dictionary, it's just to describe a branch. Okay, it's just to describe branch. Sometimes it's the bigger branch, sometimes it's the sub branch. You can always use that Lin concept to describe the branches, no matter if it's a root branch or it is an end branch. It really doesn't matter. So on this slide, when they say it's a Lin negative, that means the CD44 cells eventually will differentiate toward some lineages. So these cells are Lin negative. That means they don't have, this stem cell don't have the markers of the differentiated cells in a particular lineage. Go back to the slide here. I would say, okay, let's say just focus on this part. I would say G is a stem cell marker. Maybe the G is a stem cell marker because For example, like these 2 cells here are stem cells, so the G is a stem cells marker. So the stem cell should be G positive, but it can't be, for example, B positive. Then I would say the B is a lean marker. The B is a lean marker because the B only appear in the lineage, not in the original cell.
So if B is the marker, would there be a positive or negative sign next to that, or?
It's a negative sign there. The lean negative, you have a lean negative sign there. So CD44 positive, CD24 negative or low, and then lean negative.
So when you have a positive sign, does that determine that it's differentiated, or?
No, the positive sign just means present.
Oh, okay, okay.
Let's say CD44 positive, that means CD44 is present in this cell.
Thank you.
The CD24 is absent in this cell. Lean, those markers, negative in this cell.
Got it. Thank you.
Okay, good question. Good question. Okay, I will accelerate a little bit because today it's very technical, and I plan to cover a lot of things than what I do usually. And here, because when we try to isolate cell, we're often dependent on fluorescence detection methods. So I want to give you just a little bit background of fluorochrome. So there are a lot of fluorochrome, like shown on this slide, where you have Texas red, cyanin 5, all kind of names. Okay, those are fluorochrome. That means they can give rise to fluorescence if you have the right setup. What is the right setup? Basically, you need some light source to excite the fluorochrome. At baseline, they don't glue any fluorescence until you give a laser beam of a certain wavelength. You give the cell, give those fluorochrome that excitation, they will emit light in a certain wavelength. Okay, so there are 2 major, critical parameter here. One is their absorption max wavelength. The other is their emission maximum wavelength. So what that means is, for example, like this Pacific blue, the Pacific blue here, they get excited by wavelength 405, and then they emit light in 455 at their wavelength. So basically, you check their absorbance wavelengths to determine what laser beam are you going to apply to this particular fluorochrome. And then you observe the other wavelengths to decide to use what filter to capture the light. So basically, you use a light source to excite it, and then you use a filter to capture the light. I hope that makes sense, because the light is a spectrum. If you don't distinguish you don't use a filter, a lot of light will appear as white. But you only when you use a filter, then you can capture blue light and green light and such, right? So practically, why this 2 absorbance is important, because the first wavelength, absorption wavelength, will help you to decide, do we have this equipment that have the excitation light source? Because every equipment is different. Sometimes you only have one light source. For example, you have a violet laser light source, and then you cannot excite the others because that laser can only give you wavelengths around that range. Okay, that's what it means. You need a light source to excite your fluorochrome, and then you have to use a filter to capture the light. Now here, there is some practical consideration is, for example, in the previous slide, you want to detect CD44 and CD24, and then you want to label these 2 protein with antibodies, but you have to use 2 different antibodies. And now those two different antibodies will have to have a different fluorochrome conjugated to the antibody, two different ones, so that you can detect two different things, right? If those two detection will both give you green light, then you're confused. What have you you have just found? Yeah, so in this example, they use one fluorochrome called APC, the other called PE for a reason. And then in practice, you want to use two fluorochrome that's far away from each other in terms of emission. For example, you probably want to use a FITSI. Their emission peak is around 520, somewhere like here. And then the next one, you probably want to choose a PE because the emission maximum is around 650. You can see their peak are far away from each other. So they're not overlapping with each other. So that means when you use a filter to try to capture the green light, you won't capture the tail of that red light. Okay, so that's practical consideration when you want to choose two fluorochrome or three fluorochrome to detect different things. The same principle will apply to macroscopy as well as flow cytometry. Okay, so here I want to, maybe I should skip this one because it's in our textbook. You can read by yourself. Basically, the concept here is, you have this filter. This filter will only allow the light of a certain wavelength to pass through. Okay, other light will get reflected to somewhere else. It won't pass this filter. Only light of a certain source of a certain wavelength will be able to pass through. For example, like green light will pass this filter, while red light will pass another filter. So by changing the filter, you can detect a certain signal. And eventually, when you want an image, you're using the computer to recompose the image, because each channel or each filter only give you one image. That's maybe the green image. The other will give you the red image, and maybe the third filter will give you additional colors. And eventually, you can put those colors together to give you this kind of image. So you're detecting different kinds of cells with different kinds of markers by this way. Okay, I hope this is the fundamental cell biology. I have to move on. But on this slide, I want to describe a very useful technique called flow cytometry. I don't know if you have learned this in cell biology, but this is a very useful technique. So basically, the cells will have cell surface markers, like the CD markers, CD117, for example. And now you want to use CD117 labeling to get the cells detected. So how you do it, basically, you have an antibody against the CD117, but this antibody will get conjugated with some kind of a fluorescence, right? For example, green fluorescence. But at the same time, as I described at the beginning, sometimes you need to have additional labeling. Those additional labeling could be another positive labeling that gives you a double positive so that you can define a cell type, or sometimes it's one positive, then the other one will have to be negative. The other marker will have to be absent, so to define a cell population. And now, if you stain the cells with that antibody, and that antibody will carry the fluorescence, but they're not excited. The fluorochrome is there, but not excited, but now get attached to the cells. And now you allow that to pass this equipment called flow cytometer, and then you use a laser. The flow cytometer will give you a single cell stream. The cell will pass this tube one cell at a time. It's just by engineering, you can get the cells to pass at one cell at a time, and then this laser beam at some point will excite that cell, and then the light will get captured by 3 different filters. Maybe I shouldn't say filter, because it's just some device there to capture the signal. The one is called forward scattered light. The other is called side scattered light, while the third, often many, not only the third, maybe fourth, fifth, they would detect the fluorescence signal. So the flow cytometer will give you information whether that cell is positive for a certain marker, but at the same time, it will also tell you additional information, because the side scattered light and the forward scattered light will tell you the other physical properties like size, shape, optical homogeneity. I will give you an example because why I want this example, because I have explained to you that you can always capture the fluorescence, like in the previous example, the CD24, CD44 example, you can detect the 2 fluorescence. That's not complicated, very easy. But the real usefulness of flow cytometry is not only about detecting fluorescence. You have to take advantage of the forward scatter and the side scatter capturing device as well. Here, I want to give you this example. Basically, we're going to do a lymphocyte analysis. So you isolated the lymphocyte from blood, and then you pass those cells as a single cell stream, and then you captured signals. But in the first plot, what you did is plot the forward scatter, the signal captured by the forward scatter and the side scatter. That's forward scatter and the side scatter. And you can see the cells goes to different places because the cells are of different size, different shape, and different optical property. So you have different clusters. And now you can make this different cluster of cells to go through the analysis of the labeling, because the cells were labeled at the very beginning. So now you just capture the different information. Now, let's say if you circle this red group here to do the analysis, you will see the red group goes to, some goes to CD44 positive, some goes to CD8 positivity, right? But some of the red cells also becomes double negative because they're neither positive for CD8 or CD4. Okay, so, but if you group this cell type there, it's another group. They're different from R1 group. Now that we call this the R2 group, you can see this R2 group are mainly positive for CD4, but negative for CD8 because they have a higher expression of CD4 than this black cells, but they are kind of totally negative for CD8. But when you compare this R1 cell and the R2 cell, in the R1 population, you do have some population that's even stronger in CD4 expression, right? So the CD4 expression is intermediate in R2 than R1. So I guess what I'm trying to explain to you here is the flow cytometer will give you additional information than just the fluorescence, just the cell surface marker. Because apparently, if you just focus on CD4 positivity, you're going to consider, okay, this is a stem cell population. But if you also consider the forward scatter and the side scatter, you will know actually these are 2 types of cells there. It's not the same identity. You have 2 populations there. Maybe they are of different shape even. Okay, and then that will help you to understand the cells are not uniform. There will be a subpopulation. You would like to further, categorize the different populations there. Okay, so if you are interested in that, we can discuss further afterwards. But I have to go. I have to move on. On this slide, I want to describe to you that In addition to analysis, the flow cytometry can also be used for cell sorting. This is called fluorescence activated cell sorting. So basically you do the same thing. You label the cell with fluorescence antibody and then use a laser beam to excite the cells so they can display red or green fluorescence or no fluorescence at all. And then based on that information, in the next step, the equipment can apply a high voltage to those cells. Like the red cell, they give a different voltage. To the green cell, they give a different voltage, maybe even the direction of the electric field. So those particles, the cells in a droplet, will bear different charges. They're either negatively charged or positively charged. And then in the next step, they get separated based on their charge. Therefore, you can separate the red cell, green cell, or the cells that doesn't have any color. So this way, you can first label the cell to label the stem cell, and then later on, you can get them collected into an individual tube. And now you have a high degree of enrichment of the stem cells. And this is I would say, the most useful way, or the most used way in getting stem cells collected, isolated. And on this slide, we have another technique. It's called magnetic activated cell sorting, which is very similar to flow cytometry, to fluorescence activated cell sorting. But now, when we label the cell, we don't use fluorescence. The antibody is conjugated with some kind of magnetic beads. Okay, so instead of fluorescence, the antibody now carry magnetic beads to the cell surface. And then you can use magnet to get the cells captured, because the stem cell bearing some surface marker now get labeled by magnet labeling. Based on their cell surface marker, you can use a magnet now to pull those cells out. So I'll just leave it there, because that method is very similar to flow cytometry, but you use magnetic as the way to get the cells captured. I would say this is a poor man's flow cytometry, because in the previous example, you learned that flow cytometry can capture a lot more information than just the cell surface marker. But here, in this magnetic activated cell sorting, you rely on only the cell surface marker. No other additional information, important additional information, will be included in this isolation. If we're isolating the cells in the previous example, you're going to get the mixed population into the collection tube, because they have high and low expression, their cell shapes may be different, but here you can't distinguish that. You can only get the cells isolated by the presence of a surface marker. And now, on this slide, I'm going to tell you that you don't only rely on cell surface marker. Sometimes you rely on the specific or unique property of a cell membrane. For example, somebody in Baylor College of Medicine, whose name is I believe it's Goodell. It's Goodell. I believe she's still the professor there. Goodell first described the site population, because on the surface of stem cells, often they can express this protein called ABCG2. This is a channel protein. The presence of this channel protein will allow the cells to pump out a dye. This is called a Hooke's dye. Hooke's 342, which is a fluorescence dye. You can incubate the cells with this Hooke's dye, and the Hooke's dye will diffuse into the cell. They can pass the cell membrane slowly. You can get the cells labeled, but stem cells somehow, because of the presence of this channel protein, they can get this dye exported. This is called a dye efflux. They can get this dye out of the cell. Therefore, if you do a staining with this Hooke's dye, what you're going to see is, now look at the horizontal dimension is Hooke's red, and the vertical dimension Hooke's blue. That means this dye actually goes to 2 channels. This dye, the wavelength goes to the red filter as well as the blue channel. So it's the filter will, both filter will capture some signal of this dye. You can see in muscle cell, you have this so-called side population. Why is the side population? Because this population have low expression of Hooke's. The Hooke's get exported. The major population is high in Hooke's blue and high in Hooke's red. Well, the side population have lower expression of Hooke's. I shouldn't say expression, lower signal of Hooke's, right? But in the presence of verapamil, this is a calcium channel blocker. And this channel, ABCG2, is calcium, is a calcium channel. So if you use verapamil, in the presence of that, basically it will block this channel, and then it can no longer get the dye out. So all the cells now have a higher intensity of Hooke's. So this side population is a stem cell population. This not only happens in muscle, it happens in bone marrow as well. So in the bone marrow, you can use this to identify the stem cell. This is a unique property of the cells. And then on this slide, I'm glad I'm still within 1 hour. On this slide, I will tell you some additional feature of stem cells, adult stem cell. So you can use this unique feature to, again, isolate itself. As you can see in today's discussion, we'll always rely on some kind of fluorescence and flow cytometry to tell what is a stem cell, and then you can use FACS to get them isolated. And on this last slide, we'll use the proliferative feature of stem cells. So read this line, it's very interesting. So adult stem cells are less proliferative. As I said, when they're in their niche, they're often in quiescence. They don't proliferate at all, although they have super ability to replicate themselves, but the adult stem cell often is in the niche. They're not in an environment that require them to proliferate. So they're quiescent. They don't replicate. And now you can use some technique to capture those cells that are not dividing. One is called BRDU, the other is used as a substrate. So you use the same principle. Basically, in the case of BRDU, this is BRDU is an analog of thymidine. You know, thymidine is a building block of DNA. When a cell will divide, they first replicate their DNA. They make 2 copies of genetic material, then they divide, right? So if you incubated the cell with BRDU, the cell will mistake the BRDU as thymidine. They're going to take BRDU into the DNA, right? This way you can get the cells labeled. The BRDU will give you fluorescence. Oh, no, I'm sorry. The BRDU itself won't give you fluorescence, but then you can use an antibody against the BRDU. The antibody can carry a fluorescence. So the cells labeled with BRDU can be detected by fluorescence conjugated antibody. And now you can use flow cytometer to analyze the intensity of the fluorescence. Like in this example, it's not about BRDU, but rather we're using a substrate CFSE, which is a carboxyl fluorescing succinyl ester. So there's a long name CFSE, but this, once you get them into the cell, some enzyme will make this dye to glue fluorescence. Okay, now, when you culture the cell, they're going to go through proliferation. And now, if you get to the whole population through flow cytometer, you're going to see that some cells didn't divide at all. So they have the original amount of CFSE. They keep all the CFSE. But when the cells start to divide, they're the intensity will drop to half of the original, right? Because you only incubated the cell at the very beginning with this dye. Okay, the intensity will drop if the cell will divide. So by each time the cell divide, the intensity of that fluorescence will drop further and further. And this way, if you make the cells to go through a flow cytometer, you will know some cells have the highest intensity, some cells will have minimal amount of fluorescence. And you can define the numbers of cell division this way. And then you will collect the cells that have the highest intensity because those cells didn't go through differentiation, they didn't go through proliferation. So those cells are likely adult stem cells. Okay, so there are many other ways of stem cell isolation. Today, I just introduced to you mainly 3 categories. One is a cell surface marker using fluorescence techniques. The other is using unique features of the membrane, the side population cell. And third is we use the feature of cell proliferation to capture stem cells. And I briefly mentioned the using cell culture to capture the cell spheres. Okay, that's another way to do it. I'm sorry that I'm late again. So today I put too much content here, but like I said, I just to introduce to you guys the most important, most exciting techniques and concept in the field of stem cell biology. I'll try to control the time better next time. But maybe let's postpone this to week 3, but to Wednesday. But can you guys, the groups please decide maybe 4 dates instead of just 2, because as I said, we're gonna do a first come, first serve on that day. So I'm gonna call up group 1, please name a date, through group 6 for the first round. And then in the second round, I'm going to call up group 6 to name a date. And following that, reverse order to group 1. So it's kind of fair. You can pick the dates that you intend to give the presentation. So I updated this timetable because this week I'm going to do like lecture entirely. I pushed that date to the end of this semester. So groups, please decide the date. On Wednesday, beginning of the lecture, I'm going to ask you to give me the choice and then get that done Wednesday. And thank you very much. If you have questions, you can stay and ask me.
Wait, Professor, did you say we needed to do 4 dates instead of 3?
I'm sorry.
Did we need 4 dates instead of 2 dates? For the presentation?
You can pick 4 days. The reason is because, when they say when a group pick a date, you can't pick it any longer.
Oh, okay.
You should have some backup dates. That's why you pick more dates, so it's more flexible.
Professor?
Yes.
So do we pick our group members, or like you're gonna assign us with certain members?
You can find your group members already. I assigned you to one of the 6 groups.